Hello and welcome to the D 3.3 homeostasis review video. Alright, so standard 1 is the definition of homeostasis. So the IB definition of homeostasis is the maintenance of the internal environment of an organism. And so what does that mean? It means that certain variables, such as body temperature, blood pH, blood glucose, and blood osmotic concentration are all kept within limits. So if it goes outside that limit, it will shift in one direction or another. So if the value goes too high, it gets lowered. And if it goes too low, it gets raised. And so you have like this window. This is like where it should be. And if it's over here, it'll go this way, and if it's over here, it'll go this way. So we maintain these variables within a limit. This is true for not all organisms, right? But this is true for all mammals are going to be regulating all of these. And so when we say we change it like this, it leads us to standard 2, which is our negative feedback loops. So those responses we see right there are negative feedback. So if it's too high, you lower it. If it's too low, you raise it. That would be negative feedback. And it's more common because you're always trying to get it back to that set point. So you can think of it like a thermostat, where if it's too cold in the house, the heater kicks on. If it's too hot, the AC kicks on. So those are examples of negative feedback, which is much more common in homeostasis than positive feedback. Alright, standard 3. Standard 3 is all about blood glucose. How do we actually regulate blood glucose? So you have glucose in the blood, or blood glucose, and you have glycogen. And that glycogen is primarily, for right now, in the liver. We also find it like in the muscles too. But for this context, we're referring to the glycogen in the liver. Now, the blood glucose and the glycogen, you can go back and forth between them like this. And so if your blood glucose is too high, insulin will lower the blood glucose by causing it to become glycogen again. And so insulin is created by the beta cells of the pancreas, and it acts on most of your cells, but it specifically targets the liver and your muscles to help store glucose. Now, if your blood sugar gets too low, so when your blood sugar is low, then what we see is glucagon will act. And glucagon will raise your blood glucose levels. And so we can see how that's negative feedback. So glucagon will raise it. Glucagon is coming from the alpha cells of the pancreas. And so this is specifically acting right on the liver to raise the blood glucose levels by releasing that extra stored glucose that's being stored as glycogen. Remember, glycogen is 1,4 and 1,6 linkage glucose. Okay, next. Well, what if you don't regulate it correctly? if you don't regulate it correctly, you end up with diabetes. And there's two different types. Alright, there's two different types of diabetes. Type 1 is when you do not produce insulin. And this is due to, it's an autoimmune disorder where you destroy the beta cells of the pancreas. And so you can't create any insulin because you don't have any beta cells, because they were destroyed by your immune system. In type 2, you have what's like insulin insensitivity. You produce too much insulin, and so you no longer respond. So risk factors would be like family history and genetics. This one is also a family history, but it's also like obesity. Fine. So this one is like late onset. This is earlier onset. This one is preventable. This one is not preventable. Oh, well, there you go. And the preventing is like your diet, right? You need to have a good diet. And then treatment is injections. And for this one, it's diet and medicine. So we have a lot of new medicine now that can treat type 2 diabetes. It's in the news because it also helps regulate weight. Okay. Standard 5, thermoregulation. So thermoregulation is maintaining your temperature. We're actually going to do 5 and 6 together, because they're both about thermoregulation, but they're two different standards. There we go. Alright, so what do we need to know about our thermoregulation? Kind of a lot, which is why there's two standards on it. But the first thing to know is like which organ is most responsible. And that's the brain, but specifically we're talking about the hypothalamus. So the hypothalamus is super important for homeostasis in general, and especially thermoregulation. And so what it does a couple different things. So let's do on this side, let's do hot, cold, and split it like that. So if it's hot, it's going to trigger perspiration or sweating. And so that's evaporative cooling. Evaporative cooling. Cooling. So you cool by letting the water evaporate off your skin. It's also going to trigger vasodilation to bring blood to the surface to release heat. When you're cold, it'll trigger shivering or rapid muscle contraction, as well as vasoconstriction. We also get our goosebumps, the raised hairs, which don't really do anything anymore, but they used to. And then newborns use the brown adipose tissue. To do respiration, and it's specifically uncoupled respiration to make heat. So that's only in newborns. Now, there's another one too. Another thing that affects it, and that's the pituitary gland. And it's mostly for the cold, so we're going to leave it over here. The pituitary gland. So the pituitary gland and the hypothalamus are connected, but the pituitary gland will send signals, or receive signals, from the thermoreceptors. And what it does is it secretes TSH, or the thyroid stimulating hormone. That then acts on the thyroid gland and causes the thyroid gland to release thyroxine. And thyroxine acts on every cell in your body. It acts on all cells to increase metabolism. So it causes you to heat up because you're increasing metabolism, which then generates heat. So it's another way of raising your body temperature. So if you're cold, just make more thyroxine. Problem solved. Alright, that is the end of the SL standards. So HL, what do we got? Now for HL, the first 4 standards are all related. So we're going to kind of do them all at once. Alright, so the first 4 standards are all about the kidney. And so we're just going to go through them. The first one, standard 7, is just an overview of what does the kidney do. So we're going to start with standard 7, kidney. So the kidney looks like a kidney bean. It has blood in, blood out, and then junk out. So you have the renal artery, the renal vein, and the ureter. And so this is going to be waste. And then this is unfiltered blood. And then filtered blood. And so one of the main functions of the kidney is excretion of waste. And so it's taking toxins and other waste particles like urea out of the blood. And so you're going to get rid of that. It also is important for osmoregulation. Osmoregulation is the regulation of the osmotic concentration, which is a way of saying the number of solutes, right? So it's osmoles per liter is how we measure it. And so those are the 2 parts. Now inside of the kidney you have 2 parts as well. You have the outside, which is the cortex, and the inside, which is the medulla. And so the cortex is where like excretion and absorption happens, and the medulla is where osmoregulation happens. And these all are happening inside of the parts of the kidney which are called nephrons. So your kidney is made up of many nephrons, and the nephrons are what actually do the functions. So let's talk about them. Your nephron looks like this. So it has this part here. Like that. Okay. And so you have the Bowman's capsule first. Then you have the proximal convoluted tubule. Then you have the distal convoluted, well, that's not right. Then you have the loop of Henle. Alright, so you've got the loop of Henle. Then you have the distal convoluted tubule. And finally you have the collecting duct. So each of these has a function, and some of them are grouped together. So if we were to draw a line like this, everything above the line would be in the cortex, everything below the line would be in the medulla. And that's how it splits the functions. So filtration and excretion is the top part, and then the bottom part is osmoregulation. So that leads us into standard 8, which is all about excretion. It really, in my mind, should be titled filtration and excretion. But that's, what do I know? Alright, so step 1, first you have ultrafiltration. Ultrafiltration removes everything from the blood except red blood cells and proteins and platelets. Or blood cells and platelets. Because these are all too big to be removed. And how does it do this? So what you have is, you have arterioles branch into what we call the glomerulus. And so this is basically arterioles splitting into these like capillaries. And then these are going to have high pressure. And you're going to push everything out. And you're going to push it out into the Bowman's capsule. And then it will enter through the basement membrane. So it looks like this, you have this, and then it goes round, branch, 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 glomerulus, super branchy, and then the Bowman's capsule looks like this, and the basement membrane's right here. So you have Bowman's capsule, basement membrane, and then this is the glomerulus, the splitting of the arteriole. So this pushes everything in here. Now we call it filtrate. So everything's in here, everything's in the filtrate. Well, you don't want everything in the filtrate. That would be really bad. You'd just be sending everything out, peeing everything out of your body. So you need to reabsorb things. And so that comes to the proximal convoluted tubule. So that's the next part, is the reabsorption. And so the proximal convoluted tubule comes after the Bowman's capsule. It's the part that looks like this, kind of. Whoa. Why does it look like this? It's got large surface area, right? To increase reabsorption. Large surface area. And so you're going to pull most things in through active transport. And so you're going to pull it back into the cells and then into the blood. You're going to actively transport glucose, amino acids, vitamins, etc. You're going to actively transport those back into the cells and then put those into your blood. So those are all going to go back to the blood. You're also going to pull in a lot of ions and a lot of water. So you're going to pull all of this back into the blood because you want all of these things. And you're going to leave waste like urea and toxins. And those will eventually become urine. Great. So then from there what we have is we have osmoregulation. So that's excretion. So now we have osmoregulation, which is standard 9 and 10. And so there's two parts to it. There's the loop of Henle and then the collecting duct. So that's standard 9 and standard 10. The loop of Henle is in the medulla. Well, they're both in the medulla. And the medulla is very salty. What does that mean? It means it has really high ion concentration. And what this does is it causes water to leave. And so in the ascending limb, you are actively transporting out. You actively transport out ions. And on the descending loop, or the descending limb, it's permeable to water. And so the water can exit. And because it's super salty in the medulla, that will cause water to exit. And so you'll get even more water back. Then over here, it's all connected, right? And then you have the collecting duct. And so now, this is going to go and become urine. And running alongside it, you have capillaries. And so what you can do is you can pull water from here to the capillaries. And so now we're on to standard 10. So you can pull water from there to the capillaries. And this is all about maintaining water balance. And so the hypothalamus is back in play again. So the hypothalamus senses the osmotic concentration. So the hypothalamus is sensing the osmotic concentration, And if it is outside of what it should be, it tells the pituitary gland to secrete ADH. And so this is specifically when there is not enough water in the blood, and so when you need more water. So this is for need more water. You're going to secrete ADH. Now what does the ADH do? The ADH acts on the collecting duct. And so what it does is, if you have the collecting duct like this, you've got your cells right here. These are the cells. It will cause them to insert aquaporins. And so ADH causes you to insert aquaporins, so you have more aquaporins, which means more water reabsorbed. When there's no ADH, those aquaporins are stored in vesicles. Okay? So it's vesicle to membrane, and this is caused by ADH. And so that is how we control the amount of water. So the most of osmoregulation occurs in the collecting ducts. The point of the loop of Henle is to create the hypertonic and create that high solute so that water wants to move, and then the collecting duct is regulated by the aquaporins. Because remember, water primarily needs to leave through facilitated diffusion, special facilitated diffusion called osmosis. So these are the cells of the collecting ducts have these in it. Alright, the last standard is unrelated-ish, right? It's all about blood supply. And so for blood supply, what we're looking at is where does it go? And so you have like sleep, vigorous physical activity, and rest, wakeful rest. And so the brain and kidneys always get a lot. So brain and kidneys have lots of blood all the time, right? Because you've got to filter your blood. But the brain gets even more during sleep time, okay? Because that's when your brain is kind of resetting itself. Vigorous physical activity, right? So we're going to see that the muscles are going to go up and the gut is going to go down because you're running away from a polar bear. And so you need to not be wasting energy on digesting things when you're trying, and you need it all at the muscles. And so then during wakeful rest, right, your gut will go up some, and you'll have a lot at the kidneys too. You have a lot in the kidneys at night as well. Okay. And so that is our changes in blood supply.